Okay, we're back at this is part 2 of activity 8, lesson 3.2, protein, structure, and function from the deer, mouse, fur color, from the field to the beach, touch device version. We're on page 4, and we're comparing the two simulations. The protein synthesis simulation we saw on page 2, and the multi-level simulation we just saw on the previous page. We have worked with two simulations of the proteins. One is protein synthesis simulation, used on page 2, and the other is the multi-level simulation, where you inspected receptor proteins on page 3. What aspects of the receptor protein are easier to observe and think about using the protein synthesis simulation? Well, I'm helping you out here because this is a complex idea. This protein synthesis simulation shows that one amino acid is different because one codon is different. And the one codon is different because one nucleotide, one of the GATC nucleotides on DNA, produces a difference in the RNA, which is GCAU nucleotides. We can more easily see the mutation in this particular simulation, so we can see what's going on at the DNA level. In this multi-level simulation, we can see the results of the mutation. What happens when the DNA is mutated? We know that the shape changes and it doesn't bond the signal protein anymore when the mutation is present in the light mouse. So we can see that the overall shape of the protein that results from the amino acid sequence is easier to see in this simulation, in the second simulation. The protein synthesis sim does not show you the finished shape of the whole protein. It shows you, well, sort of a shape, but it doesn't show you kind of the big picture of the shape. We can more easily see the results of the mutation. What happens because the DNA is different? How can using the simulations together help us understand the differences in fur color of the mice? So I gave you a head start on this question, because using the simulations together can show us differences in the alleles. That's at the DNA level. We can see the two alleles, the differences in the DNA, and the differences in the proteins that result as a result of the mutation in the DNA. So we can see the protein level, the difference in the shape of the protein in one simulation, and we can see up close the DNA level mutation. And you can write some additional information here about how we can understand differences in the color of the mice. What are those differences and how can we see them with these two simulations? All right, let's go to the next page. We're on page 5. Connecting the biology from a mouse's DNA to its fur. The DNA that codes for the receptor protein is from a gene called MC1R. The MC1R DNA in the dark brown mice is slightly different from the MC1R in the light brown mice. So that's in the gene that codes for the receptor protein. You found a difference in the DNA on page 2 of this activity, where you saw that the dark brown mice had a C where the light brown mice had a T. Those are the nucleotides in the DNA. These two forms of the gene are known as alleles of the MC1R gene. An allele is a variation of the DNA sequence of the gene. They're kind of like versions or flavors of a gene, like chocolate and vanilla ice cream. They're both ice cream, they're just a little different. We refer to these as MC1R alleles as R light and R dark, or R L and R D for short. The R in the allele name is from the receptor protein. You also saw that the differences in the DNA cause a difference in the chain of amino acids that forms the receptor protein. The dark brown mouse has an amino acid called arginine, where the light brown mouse has cysteine in the same position in the protein. Those are the amino acids that are a result of reading the DNA, which is transcribed, or reading the RNA, which is transcribed from the DNA. So I've already completed the table here. We know the differences are C versus T in dark versus light, and this is at the DNA level. That's transcribed to RNA, which is then read in the ribosomes outside in the cytoplasm, which assembles the amino acids. There's a different codon, as you can see, for each of these mice. You can see the difference here. And one codon gives arginine and the other gives cysteine in that position, that the 67th amino acid position. The symbol here is MC1R dark and MC1R light. The fur color words are dark and light. So here's our summary table that helps us realize the DNA level differences that are translated into the protein level differences. Let's bring it all together. Refer to the table and summarize how the DNA for the MC1R gene and the mouse influences fur color. I'll give you a word bank here to make your job easier. So we know we have DNA differences, that they're transcribed into RNA differences that results in codon differences, and that's which result in amino acid differences. The amino acid sequence is different, and so the protein folds differently, which results in a different shaped protein. So there you go. That is not the answer. That is a word bank for getting started on the answer. All right. You can pause the video, finish your answer, and then we'll go to the next page. So this is page 6. We're doing the model of fur color. We've looked at how the light brown and dark brown mice differ in their nucleotides and amino acids. Find and review the models for light and dark brown mouse fur color that you developed in Unit 2 and updated in Lesson 3.1. With your revise your 3-point model to explain the relationship between the mouse's DNA and the color of its fur. Make sure to include the steps that connect DNA, the function of the receptor, and fur color. Use evidence and ideas from the simulation. If you need to make many changes, it might help to start on a fresh piece of paper to take a picture of your revised model. So we'll go back to look at the model that I showed you at the beginning in part 1 of this 3.2 lesson. Just so you know, I pulled all of my icons, all of my pictures from this website. It's connectedbio.org/icons. You can copy and paste these icons or do a save as and save the image file. There's lots of different icons that will be very helpful in explaining how the cell makes eumelanin or pheomelanin and the different colored mice. So here's the model that we were looking at part 1 of lesson 3.2. And this is not a complete model. It needs a lot of explanation. It needs some arrows. It needs some words explaining how these icons are related to each other, how things are different, how we get from this RNA to the protein, why one protein works, why this protein bonds signal protein and it ends up making eumelanin, and this receptor protein does not bond the signal protein, does not make eumelanin here, and just makes pheomelanin. So it's your job, if you want to go to this, I'll give you the link to this Jamboard, in the notes for this YouTube. You can go there and make your own copy and label it, or you can take a screenshot and you can label it in Notability. So once you've finished your model, you can upload a picture. If it's on paper, you can just take a photograph. If you're using an app, a drawing app, you can just save it as a JPEG and upload that image. For question 22, we're going to return to page 1 of this lesson where I made a prediction about how the receptor proteins in the dark brown and light brown mice are different. So how does your current model add to, agree with, or contradict your prediction? I gave you a word bank here to get you started on explaining what's going on. There's different nucleotides in the DNA that are transcribed to the RNA. They go to the amino acid that ends up in a different amino acid. When you have a different amino acid, you're going to get a different shaped protein. That different shaped receptor protein doesn't connect to the signal protein, those blue globs, to break it apart. There's no activated signal protein, so no dark pigments created. If you don't have dark pigment, you end up being a light mouse. So that's probably not how your current model agrees with or contradicts your prediction, but that's a good explanation of what's going on. We can use this explanation when we go to lesson 3.2, table 1 in our field notebook. So here's the lesson 3.2, table 1 summary. What did we learn? Well, what did we do? We transcribed and translated DNA. DNA of dark and light mice. And we also found the amino acid difference. And we discovered the shape change as a result of the mutation in the light mouse. What did we observe? What patterns did we notice? Well, we could say when there is a mutation, there are changes at different levels. What have we figured out? Well, mutation in the light mouse DNA makes the protein function differently in light mice. We'll be more specific here and say receptor protein. Now, this is not the answer, but this is a good start on your answer. How does this help answer our question? Well, our question is, what's the DNA, what is the actual cause of the difference between light mice and dark mice? And now we know it's a difference in the DNA that causes a difference in protein function, and that results in a different color. Okay, so that's a good start on lesson 3.1, table 1, 3.2, table 1, the summary table. And this is going to help us form our answer for when we go to the next page here, where there's a summary lesson, and we're just going to jump right into that summary lesson. And I've put a word bank here for you. You can write an individual response, and here is a word bank. You want to mention a DNA mutation. You can mention transcription to RNA. That results in a different receptor protein. There's a different codon. It picks out a different amino acid. The signal proteins aren't utilized. There's no eumelanin in the light mouse. The light mouse ends up being a different color because there's no eumelanin, because that receptor protein isn't working with the signal proteins anymore. And we're really interested, we're trying to pull everything together here. Given what we know about predators, the environment, mouse cells and proteins and mouse DNA all work together to influence mouse color. So we have to go back to the hawks. Why are these mice different? The hawks eat dark mice on the beach, and more white mice survive to reproduce. So this may have been a mutation in one mouse, but it ended up proliferating, or there ended up being a lot of white mice after a time because white mice survived and darker mice didn't, because the hawks on the beach saw those dark mice more easily and ate them. And if you've been eaten and you're dead, you can't reproduce. So that's it. Thanks for watching, and next time we'll do lesson 4.1.